Hi, my name is Hannah Persson, and I'm going to be talking about the rabies virus. So the question for this communication challenge was basically to describe what the viral life cycle is like, and why can't a virus replicate without a host cell. And so I'm just going to take us through basically the steps of the viral life cycle in terms of rabies. And so that first step is going to be basically cell entry. And so how the rabies virus does this is it goes through a process called endocytosis. And what that basically is it's just a way that any virus can enter a cell. Endocytosis isn't restricted to membrane-bound or non-membrane-bound viruses. And so this is basically just the method that the rabies virus utilizes. There are other methods. There's membrane fusion, but since that's not how the rabies virus enters the cell, we're just going to focus on endocytosis. And so I have this diagram here that I drew on the side that basically maps out the entire process of endocytosis. So you can see at first the virus is just floating around outside the cell, and then the virus creates kind of like an infolding around the host cell's membrane. And in the third step, you can see the membrane-bound virus is kind of just floating around inside the cytoplasm. And then the membrane deteriorates, and the genetic material that was trapped inside of the virus is also released into the cell. All right, so the next sort of part of this is what does this genetic information do once it's successfully released into the cytoplasm of the host cell? And so I've separated this into 5 basic steps. And the first step of this is going to be transcription inside the cytoplasm, because the RNA inside of the virus is single-stranded and negative sense. And so what that means is that it basically has to undergo transcription before it can be replicated or do protein synthesis or any of that. And so it undergoes transcription in the cytoplasm. And then in the second step, the transcribed RNA has it sort of has to pick and choose whether or not it goes to the nucleus for replication, or if it travels off into the cytoplasm and carries on with protein synthesis. Once this has all sort of happened, all of these new virus particles travel down, and they kind of gather in the endoplasmic reticulum, and a vesicle is formed, and it travels around in the endoplasmic reticulum, and then it's going to continue on to travel through the Golgi apparatus, and in there it's going to fuse. The virus proteins are going to become modified a little bit, and then the vesicle is going to travel to the cell, the outer cell membrane, where it's going to deteriorate, and then the virus is going to be released into the body, where it's going to travel around and try to infect other host cells. And so, this is basically the entire process of the viral life cycle for rabies.